You know, for the longest time, editing our own DNA, that was pure sci-fi, right? Something straight out of a movie. But then came CRISPR. This incredible technology didn't just blur the line between fiction and reality, it completely erased it. And what's wild is that the COVID-19 pandemic, well, it basically hit the fast-forward button on all of this, pushing innovation in ways nobody saw coming. And the whole story? It kicks off with a really fascinating question.
I mean, just think about that for a second. What if our best defense against a brand new, terrifying virus was actually hiding inside ancient bacteria? It sounds kind of crazy, right? But this isn't some what-if scenario. This is the actual origin story of a tool that is literally changing our world.
Okay, let's just go back in time for a minute. Remember the early days of 2020? The world was just trying to get a handle on this new virus that was spreading like wildfire. And to even have a prayer of slowing it down, we needed one thing, and we needed it badly: a way to test people. Not just any test, but one that was fast, accurate, and available to everyone.
So, the problem was, our main tool, the so-called gold standard, RT-PCR, was, well, it was good, it was accurate, but it was also a massive bottleneck. You see, it's a complicated lab test. It took hours, sometimes even days to get a result. Plus, you needed all this expensive gear and specialized technicians. We needed the exact opposite. We needed something fast, cheap, and simple enough to use just about anywhere, like a local clinic or even a small town. Basically, the world was crying out for a diagnostic revolution, like yesterday.
And that's where our hero, CRISPR, steps onto the stage. Now, the coolest thing about CRISPR is that it's not some brand new gadget cooked up in a lab. Nope, it's this brilliant, ancient system that nature has been using for eons. Scientists just figured out how to borrow it. The best way to think about it, it's like nature's very own, super precise Google search for genes.
So what is this thing? At its core, CRISPR is basically an immune system for bacteria. I mean, for literally billions of years, bacteria have been in this constant war with viruses, and CRISPR is their secret weapon. When a virus attacks, the bacteria grabs a little piece of its DNA and stores it. It's like creating a tiny most wanted poster. That way, if that same virus ever shows up again, the bacteria can spot it instantly and just chop it up.
So scientists, in their brilliance, figured out how to hijack this whole search and cut mission for our own purposes. And the concept is actually beautifully simple. First, you've got something called a guide RNA. Think of it like a GPS. You can program it with the exact genetic address you're looking for. This guide RNA then leads a special protein, a Cas enzyme, right to that target. And what does the Cas enzyme do? Well, it's basically a pair of molecular scissors. It gets there and snip. It makes a perfect, clean.
And let me tell you, this wasn't just some small discovery. The power, the sheer elegance of this system was so game-changing that its pioneers, Emmanuel Charpentier and Jennifer Doudna, won the Nobel Prize for it. We're not talking about a small step forward here. This was a massive quantum leap for all of biotechnology.
Okay, so Nobel Prize-winning tech, that's great, but how does a gene editor help fight a pandemic? Well, here's the key. Before you can cut or edit anything, you first have to search and find. And that search function in CRISPR, it is unbelievably precise. Scientists had this light bulb moment. They realized they could program that GPS to look for one very specific thing, the genetic signature of the COVID-19 virus.
And that one idea just unleashed this explosion of innovation. All these different research teams jumped on it, creating platforms with these really clever names, like SHERLOCK and Detector, and the results were just wow. Instead of waiting hours or days for that old qPCR test, these new CRISPR-based tests could give you an answer in under an hour. Some could even do it in less than 30 minutes. They were built from the ground up to be fast and easy to use.
And here's the most brilliant part, the part that really shows you what a game changer this was. You can forget about all those complex, expensive lab machines. For a lot of these new tests, the results showed up on a simple little paper strip, just like a pregnancy test. You know, 1 line for negative, 2 lines for positive. It was so simple, so familiar. It meant you could finally take testing out of the specialized lab and bring it right to where people actually work.
So, let's just recap the huge advantage here. These CRISPR diagnostics, or CRISPR-DX as they're called, they checked all the boxes. They were super fast. We're talking minutes, not hours. They were incredibly sensitive, meaning they could pick up even the tiniest trace of the virus. They were also extremely specific, so you got way fewer false positives. And the big one? They were accessible. You didn't need a huge lab anymore.
But, you know, just finding the virus, that was really only the beginning. That was just using the search function.
The real mind-blowing promise of CRISPR has always been in its cut function, the power to actually go in and edit genes. And that power is taking us from just detecting diseases to actually treating them.
So scientists started working on CRISPR-based therapies, CRISPR-TX. For COVID, one of the coolest ideas was a system they nicknamed Pac-Man. I love that. The idea was to send it into our own cells to act like a tiny antiviral machine. It would hunt down the virus's genetic code, its RNA, and just gobble it up, chopping it into useless pieces. It would stop the virus from making copies of itself dead in its tracks.
Now, don't get me wrong. Fighting viruses is a huge, huge deal. But the true, ultimate power of CRISPR, it goes so much deeper than that. It's about having the ability to fix the genetic typos that are written into our own DNA, the ones we're born with.
And I have to say this again, this is not science fiction. This is happening right now. In what was just a monumental moment for all of medicine, the FDA gave the green light to the very first CRISPR therapy. And it wasn't just a treatment, it was designed to cure sickle cell disease, this awful, painful genetic blood disorder.
Just look at this number for a second. 93.5%. In the clinical trials, that's the percentage of patients who are completely freed from the horrible, debilitating pain crises that had basically run their entire lives. This isn't just about managing symptoms anymore. This is about fundamentally changing and saving lives.
Okay, so as absolutely mind-blowing as this is, we have to remember we are standing at the very, very beginning of a whole new era in medicine. Going from one incredible approved cure to making this kind of treatment available for lots of diseases, well, that road is long and it's full of some really big challenges.
And this right here, this is probably the single biggest question, the biggest hurdle the whole field is facing right now. See, with something like sickle cell, you can take a patient's cells out of their body, edit them in a lab, and then put them back in. But for most genetic diseases, you can't do that. So how in the world do you get that CRISPR system, the guide RNA and the scissors, to only the right cells deep inside the human body? Like, how do you tell it to only go to the liver and nowhere else?
That's what scientists call the delivery dilemma. And one of the most exciting solutions is actually something you probably already know about, thanks to the COVID vaccines: lipid nanoparticles, or LNPs. They're basically these tiny fatty bubbles. They act like a delivery truck, protecting the precious CRISPR cargo inside from our immune system. And this is the really clever bit. Scientists are figuring out how to stick molecules on the outside of these bubbles that act like a zip code, telling that delivery truck exactly which organ in the body is its final destination. And even if we solve the delivery problem, there are still other big hurdles. First, there's safety. We have to be 100% sure those molecular scissors only cut where they're supposed to, and don't make any accidental, off-target snips somewhere else in our DNA. Then, there's the cost. These cures could cost millions of dollars. How do we make them affordable so that everyone who needs them can actually get them? And finally, of course, you have the huge ethical questions. When we have the power to rewrite our own genetic code, it forces us to have a really serious conversation about what that means and where we draw the line.
You know, for decades, science has been focused on learning how to read the book of life, our genetic code. But now, with CRISPR, we're learning how to write in that book. We can go in and fix the typos that cause disease, and maybe, just maybe, even write entirely new sentences. It's a technology with almost unimaginable power and promise. And the story of what we, as a species, choose to do with it, well, that story is really just beginning.